Welcome to Biomindscape. Today we are going to know. So let's begin the chapter of molecular basis of inheritance. So what is an inheritance? So inheritance refers to the process where the traits of the characteristics are passed down from the parent to the offspring, and it is essential for the transmissions of the genetic information which encode the DNA from one generation to the next, and this genetic information determine various aspects of organism appearance, behavior, and eventually susceptible to certain diseases. So genetic material is a blueprint for inheritance which carry out the form of DNA, that is the deoxyribonucleic acid. The chromosome, the DNA is organized into the structure called chromosome, which are present from the parent to the offspring during reproduction, and gene are specific segment of DNA that code for a particular trait, and trait are the observable character of an organism such as eye color, height, then hair color, which are susceptible to certain diseases. And in sexual reproduction organism, each parent contribute half of its genetic material, which results in the offspring with a unique combination of traits. So asexual reproduction, where a single parent produce offspring and that are genetically identical or nearly identical to itself. And variation is while the inheritance ensure the passing of a trait, and it also allow for the variation within a population due to genetic recombination and mutation, which can drive evolution. At time of Mendel, the nature of those factor are known as the genes, which regulate the pattern of inheritance that was not clear at that time. Over the next 100 years, the nature of the genetic material was investigated, and the DNA structure as in genetic material was obtained. So you have learned about the nucleic acid. They are the polymers of nucleotide, and DNA or RNA are the two type of nucleic acid. They are found in living system. DNA act as a genetic material in most of the organism. RNA, though it also acts as a genetic material in some viruses, but mostly as a messenger. So RNA has an additional role as well. It functions as an adapter structure in some cases of catalytic molecule. The structure of nucleotide, you know that how the nucleotide looks like. So the way these monomeric units are linked to form a nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, and the process of making RNA from the DNA, that is the transcription, and the genetic code that determine the sequence of amino acid in protein, the process of protein synthesis, that is the translation, and elementary base of their regulation. So the nucleotide sequence of human genome during last decade has seen a new era of genomics. In the last section, the essential of human genome sequence and its consequence will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, DNA. DNA is a long polymer of deoxyribonucleotides. The length of the DNA is usually defined as a number of nucleotides or a pair of nucleotides referring to a base pair which are present in it. It is also characteristics of an organism. We know that a bacteriophage, it has how many number of nucleotides? 5386. Like theta into 174, a type of bacteriophage. Then bacteriophage lambda, it has 48,502 base pairs. Each, for example, E. coli, it have 4.6 into 10 raised to 6 base pairs. Now these are some of the important points the number of base pairs in what organism, how many base pairs are present. In, for example, in bacteriophage lambda or these examples which are given in NCERT, they are important. Then E. coli, it has 4.6 into 10 raised to 6 base pair, and a haploid containing human DNA, which consists 3.3 into 10 raised to 9 base pair. Now let's know first the structure of polynucleotide chain. So we know that there is a phosphate group which is attached in between all of these chains, and a hydroxyl group at the 3' end and a phosphate group at the 5' end is given in the figure. So what is the DNA? It is a long polymer of deoxyribonucleic acid. So the length of the DNA is usually defined as a number of nucleotide. These are the nucleotide to the base pair present in it, and they are the characteristics of an organism. So a nucleotide has 3 components: a nitrogenous base, a pentose sugar, which is ribose in case of RNA, and deoxyribose in case of DNA, and a phosphate group. So there are 2 types of nitrogen base which are known as purine and pyrimidine. So purine consists of adenine and guanine, and pyrimidine consists of cytosine and thymine, and uracil in the case of RNA. So the cytosine is common for both DNA and RNA, and thymine is present in DNA, and uracil is present in RNA in the place of thymine. So a nitrogenous base is linked to the OH, that is hydroxy group, at the 3' here. It is 1 carbon pentose sugar through N-glycosidyl linkages to form a nucleoside, such as adenosine and deoxyadenosine. So when a phosphate group is linked to the OH, that is a hydroxyl group of the 5' carbon of a nucleoside through a phosphodiester linkages, so at that point a corresponding nucleotide of deoxynucleotide depend upon the type of sugar present. It is formed. So two nucleotides are linked through the 3' 5' phosphodiester linkages to form this dinucleotide. More nucleotide can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has one end and one free phosphate moiety at a 5' end of a sugar, which is referred as the 5' end of the polynucleotide chain. Similar at other end of a polymer, the sugar has a free OH of 3' carbon group, which is referred to as 3' end of a polynucleotide chain. So the backbone of the polynucleotide chain is formed due to the sugar and the phosphate. So the nitrogenous base linked to the sugar moiety projects from the backbone, as we can see in the figure. In RNA, every nucleotide residue has an additional hydroxy group, which is present at the 2' position in the ribose. So also in RNA, the uracil is found at the place of thymine, that is 5-methyl uracil, another chemical name for thymine, and DNA is an acidic substance present in nucleus was first identified by Friedrich Miescher in 1869. So he named it as a nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained for a very long time period. So it was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by the Wilkin and the Franklin, proposed a very simple but famous helix model, that is the structure of DNA. So one of the hallmark of this proposition was base pair between the 2 strands of polynucleotide chain. However, this positions was also based on the observation of Chargraph, and that was of a double-stranded DNA, and the ratios between the adenine and thymine and guanine and cytosine, they are the constant and equal. So base pair confers a very unique property to the polynucleotide chain. They are said to be complementary to each other, and therefore the sequence of the base is one strand is known, then the sequence in the another strand can be predicted. So also if the strand of the DNA Let's call it as parental DNA. It acts as a template for the synthesis of a newly strand and the two double strand DNA. Let's call them as a daughter DNA, thus produced and would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA become very clear. So the silent features of DNA helix structure are as follows. So here these are the important points which are given about the silent features of the DNA helix structure. So in the previous paragraph we have known about the base pair which confer a very unique property of polynucleotide chain. So which are complementary, and the sequence of the base in one strand is known, and which also predicts the other strands, and if each strand of the DNA is a parental DNA which act as a template for synthesis for making the new strand. So two double strand DNA we call it as the daughter DNA, and thus they produce identical to their parent DNA molecule. Because of this, genetic implications of the structure and it becomes very clear. Now other than that, we have known about the DNA as an acidic substance present in the nucleus, and the names of the scientists and their dates where he named is nuclein. Who named it nuclein? The Friedrich Waikar in 1869. So these are some of the important points, and the names and X-ray diffraction data produced by which scientist, and it is very famous because of the which model, like double helix model for the structure of DNA. Then hallmarks, what are the hallmarks? The base pairs between the two strands of polynucleotide chain, and how this propositions was also based on the observations of the ratio, who identified the ratio of adenine and thymine and guanine and cytosine. All these are the important points of this particular paragraph. So let's know the silent features of the double helix structure of DNA, which is made up of two polynucleotide chain, where the backbone is constituted by sugar phosphate and the base project inside. So the two chain have anti-parallel polarity. It means one of the chain has a polarity of 5' to 3' and other has the opposite, that is 3' to 5'. So this base in two strands are paired through hydrogen bond, forming base pairs, and adenine from two hydrogen bond with thymine from opposite strand and vice versa. Similarly, guanine bind with cytosine with 3 hydrogen bond. This is also important, that how many base pairs are present between adenine and thymine, and guanine and cytosine have 3 base pairs, whereas adenine and thymine have 2 base pairs. This is very important point in the silent features of DNA double helix structure. Then other than that, as a result, the purine comes opposite to pyrimidine. So this is also important, which pair come opposite to which one. So it have a uniform distance between the two strands of helix. We can see the diagram how it is. The two chain are coiled in right-handed fashion, and the pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of the meter, and that is 10 raised to minus 9 meter. So, and there are roughly 10 base pairs in each turn. So this is one more important silent feature. Each turn consists of total 10 base pairs in between here. So the distance between a base pair and a helix is approximately 0.34 nanometer. The plane of one base pair stacked over the other in a double helix. So this is addition to the hydrogen bond, which confirms the stability of the helical structure. Comparing the structure of purine and pyrimidine, you can find out what is the distance between the two polynucleotide chain in DNA, and remains almost constant. So the proposition of the double helix structure for DNA and its simplicity in explaining the genetic implications become revolutionary. Francis Crick proposed the central dogma of molecular biology, which stated that the genetic information flows from the DNA to RNA and RNA to protein. So by DNA, by transcription makes a mRNA, and by translation it makes the protein. So in some viruses, the flow of information is in reverse direction from RNA to DNA. So you can suggest them the names. Next topic is packaging of DNA helix. Now you know what is a histone, what is a histone octamer or nucleosome. So the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure of nucleosome. For that we must know what is a histosome or what is a histosome octamer. So in diagram we can see here there is a H1 histone present, a histone octamer, it is circular, present here. In eukaryotes this organism is much more complex and there is a set of positively charged base protein, which these proteins are called histone. So its protein acquire charges depending upon the abundance of amino acid residue with charged side chain. So histone are rich in basic amino acid residue like lysine and arginine. So both amino acid residue carry positive charge in their side chain, and histone are organized to form a unit of 8 molecule called histone octamer. Because the 8 molecule are present, so these are forming an octamer. So the negatively charged DNA is wrapped around the positively charged histone octamer to form the structure called nucleosome. And a typical nucleosome contains 200 base pairs of DNA helix, and nucleosome consists the repetitive unit of structure in nucleus. It is called the chromatin, and the thread-like strain, the colored body seen in the nucleus, is a nucleosome. In the chromatin are seen on a beads on the string in the second picture under the electron microscope. So distance between the consecutive base pairs is 0.34 into 10 raised to minus 9 meter. If the length of the DNA double helix is typically mammalian cell, it is calculated simply by multiplying the total number of base pair with distance between the two consecutive base pairs, that is 6.6 into 10 raised to 9 base pairs into 0.34 into 10 raised to 9 pi base pair, which comes out to be 2.2 meter, a length that is far greater than the dimension of a typical nucleus, which is approximately 10 raised to minus 6 meter. So such long polymer packaging in a cell you can see, and in prokaryotic such as E. coli, though they do not have a defined nucleus, the DNA is not scratched through the cell, but DNA being negatively charged is held with some protein and that have a positive charge in region termed as the nucleotide. So DNA as a nucleotide is organized in a large loop held by the protein. Then non-histone chromatin protein. It is a typical nucleus. Some region of the chromatin are loosely packed, strain light, and referred to as euchromatin, and the chromatin that is much more denser packed, and the strain is dark and it is known as a heterochromatin. So euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive one. Then the search for the genetic material. Even though the discovery of nuclein by the Meselson and the propositions for the principle of inheritance by Mendel were almost the same, but the DNA act as a genetic material took a long to discover and proven. By 1926, the quest to determine the mechanism for genetic inheritance has reached the molecular level. Previous discovery by George Mendel and Walter Sutton and Thomas Morgan and numerous other scientists has narrowed the search to the chromosome located in the nucleus of most the cell, but the question was what molecule was actually the genetic material has been answered. So the transforming principle. In 1928, Frederick Griffith is a series of experiments with Streptococci pneumonia, the bacteria which is responsible for pneumonia, witnessed a miraculous transformation in a bacteria. During this course of his experiment, a living organism, bacteria, has changed in physical form. When the Streptococcus pneumonia, that is a bacteria, and growing on a culture plate, some product smooth shiny colonies that are S, while the other produce the rough colonies R. So this become a S strain bacteria which have a mucus polysaccharide coat while R strain does not, and mice infected with the S strain virulent die from pneumonia infection by mice infected with the R strain do not develop pneumonia. So S strain and R strain when injected in mice, S strain when injected in mice the mice died, whereas R strain when injected in mice the mice lived. So Griffith was able to kill the bacteria by heating them, and he observed that the heat killed S strain bacteria was injected in the mice did not kill them. So when he see the S strain heated killed injected in the mice lived, whereas X strain heated killed plus R strain live was injected in the mice died. So injected a mixture of heat kill S and live the live R bacteria, the mice died. Moreover, he recovered living with the S bacteria from the dead mice. So he concluded that the R strain bacteria has somehow been transformed by the heat killed S strain bacteria. So some transforming principle here it formed that heat killed S strain has enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transform of the genetic material. However, the biochemical nature of genetic material was not defined from his experiment. So biochemical characteristics of transforming principle prior to the work of the Avery and McLeod and McCarthy in 1933 to 44. So the genetic material was thought to be the protein portion of the part on the work to determine the biochemical nature of transforming principle in Griffith's experiment. They purified the biochemical protein, DNA and RNA, etc. from a heat-killed S cell to see which one could transform the live R cell into the S cell, and they discovered that the DNA alone from the S bacteria caused R bacteria to become transformed. So they also discovered the protein digesting the enzyme protease and RNA digesting enzyme RNase and did not affect the transformation. So the transforming substance was not a protein or RNA. Digestion was with the DNAs did inhibit the transformation, suggesting that the DNA caused the transformation, and they concluded the DNA is a hereditary material, but not all biologist was convinced. So the genetic material in DNA. So the genetic material in DNA. The bacteriophages attaches to the bacteria and its genetic material and then enter the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particle. So there is an experiment, that is Hershey and Chase work to discover whether it was a protein or a DNA, and it formed the virus that enters the bacteria. They grew some virus on a medium that contained the radioactive phosphorus and some other on medium that contained radioactive sulfur. Then virus grew in presence of a radioactive phosphorus which contain a radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. So similarly, virus grew on radioactive sulfur containing radioactive protein but not on radioactive DNA, because DNA does not contain sulfur. Radioactive phage where they allow to attach the E. coli bacteria, then as an infection it proceeds and the virus coat were removed from the bacteria by agitating them in a blender. So the virus particle were separated from the bacteria by spinning them in a centrifuge. So the bacteria which was infecting the virus that had radioactive DNA were radioactive, indicating that DNA was a material that passes from the virus to the bacteria. Bacteria that were infected with virus that had a radioactive protein were not radioactive. This indicates that the protein did not enter the bacteria from the virus. So DNA is therefore the genetic material, and this is passed from virus to the bacteria. Now let's note the properties of genetic material, that is DNA versus RNA. From the foregoing discussion, it is clear that the debate between the protein versus the DNA as the genetic material was unequally resolved from the Hershey and Chase experiment, and it became an established fact that the DNA that act as a genetic material. However, it subsequently become clear that in some virus, RNA is a genetic material. For example, tobacco mosaic virus, then QB bacteriophage. In that, the RNA it acts as a genetic material. So answer to some of the questions such as why DNA is a predominant genetic material whereas RNA perform dynamic function of a messenger, and adapter that has been found from the difference between the chemical structure of a two nucleic acid molecule. So can you recall the two chemical difference between DNA and RNA? So a molecule that can act as a genetic material must fulfill the following criteria. It should be able to generate cell replication, that is a replica. So it should be stabilized, a chemical and structurally it should be stabilized. It should provide the scope of slow changes, that is a mutation that are required for evolution. Then it should be able to express itself in a form of Mendelian character. If one examines each requirements one by one, because of the rule of the base pairs and complementary, both the nucleic acid like that is the DNA or RNA have an ability to direct their duplication, and the other molecule in the living system such as the protein fails to fulfill this criteria itself. So the genetic material should be stable enough not to change with different stages of life cycle, age, or with change in the physiology of an organism. So stability as one of the property of genetic material was an evidence in Griffith's transforming principle itself, that heat which killed the bacteria at least did not destroy some of the property of the genetic material. This now can easily be explained in light of the DNA, that the two strand being complementary if it is separated by the heating comes together when appropriate conditions are provided to them. So further, the 2OH group present at very nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. So RNA is also known as to be catalytic, hence reactive. So therefore DNA chemically is less reactive and structurally more stable when compared to the RNA. Therefore among the two nucleic acid, the DNA is better genetic material. In fact, the presence of the thymine at a place of uracil also confirms an additional stability to DNA. So both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate. Consequently, viruses have RNA genome and having a short lifespan mutated and evolved faster. So RNA can be directly coded for the synthesis of protein, hence can easily express the characteristics. DNA, however, is dependent on RNA for synthesis of protein. The protein synthesis machinery has evolved around the RNA, and the above discussion indicate that both RNA and DNA can function as a genetic material, but DNA being more stable is preferred for storage of the genetic information. For the transmission of genetic information, RNA is better. Now let's know what is RNA world. For a foregoing discussion, an immediate question becomes evidence, which is the first genetic material. It shall be discussed in detail. So RNA was the first genetic material. There is not enough evidence for suggesting the essential life processes such as metabolism, translation, splicing, which are evolved in RNA. RNA used to act as a genetic material as well as catalyzed, and there are some important biochemical reaction in living system that are catalyzed by RNA catalyst and not by the protein enzyme. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with a chemical modification that made it more stable. So, DNA being double-stranded have a complementary strand further resisting change by evolving a process of the repair. So, replication. While the DNA helix, double helical structure of the DNA by Watson and Crick, had immediately proposed a scheme of replication of DNA to quote their original statement that it is as follows. It has not escaped our notice that the specific pairing we have postulated immediately suggested a possible copying mechanism for genetic material, which was given by Watson and Crick in 1953. So the scheme suggests that the two strands would be separated and act as a template for the synthesis of new complementary strand. After the completion of replication, each DNA would have one parental and one new synthesized strand, and this scheme was termed as semi-conservative DNA replication. So there was an experimental proof. That was the DNA replication in semi-conservatively. It was showing that the first the E. coli and subsequent in higher organisms such as a plant and a human cell, the Matthew Meselson and Franklin Stave performed the following experiment in 1958. They grew a E. coli in a medium which contained NH4Cl, that is NH is a heavy isotope of nitrogen, and as only nitrogen source for many generations, this result was that the N15 was incorporated into newly synthesized DNA as well as another nitrogen containing compound, and this heavy DNA molecule could be discussed from the normal DNA by centrifugation in a cesium chloride CaCl density gradient. Please note that N15 is not radioactive isotope and it can be separated from N14 only based on the densities. So this was some of the important paragraph which was given in NCERT, and about it, it is like that these were transforming cells into the medium with normal NH4Cl, and there were various samples that were taken, defined in various time interval in the cell, which was multiplied and extracting the DNA that remained the double-stranded helix. So these various samples were separated independently on the CaCl gradient to measure the density of the DNA. So thus the DNA that was extracted from a culture one generation after the transforming of N15 to N14 medium, that is after 20 minutes, the E. coli started dividing in 20 minutes, and it has a hybrid or a intermediate density. So DNA extract from the culture after another generation, that was after 40 minutes, that the second generation was composed of equal amount of this hybrid DNA and of the light DNA. So if E. coli was allowed to grow for 80 minutes, then what would be the proportion of the light that hybridizes the density of DNA molecule? Very simple experiment involved use of radioactive thymidine to detect the distribution of newly synthesized DNA in the chromosome was performed on Visia faba, that is a faba beans, by the Taylor and his colleagues in 1958, and this experiment proved that the DNA's chromosome also replicates semi-conservatively. So the machinery and its enzyme. In living cells such as the E. coli, the process of replication requires a set of catalytic enzyme. The main enzyme is referred to as DNA-dependent DNA polymerase, since it is used as a DNA template to catalyze the polymerization of the deoxynucleotide, and these enzymes are highly efficient enzymes as they have to catalyze the polymerization of a large number of nucleotide in a very short time. So the E. coli has only 4.6 into 10 raised to 6 base pair compared with the human, with whose diploid content, that is 6.6 into 10 raised to 9 base pairs, completes the process of replication within 18 minutes. That means the average rate of polymerization has been approximately 2000 base pair per second. Not only these polymerases have been to be fast, but they also have a catalytic reaction with higher degree of accuracy. So any mistake during replication would result into the mutation. Furthermore, energetically replication is very expensive process. Deoxyribonucleotide triphosphate serve dual purpose. So in addition to acting as a substrate, they provide energy for polymerization reaction. The two terminal phosphate in a deoxynucleotide nucleoside triphosphate are highly energy phosphate, and same as in case of the ATP. So in addition to DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of the DNA cannot be separated in the entire length due to very high energy required, the replication occurs within a small opening of the DNA helix, which we can call it as replication fork. So DNA dependent DNA polymerase catalyze the polymerization of only in one direction, that is 5' to 3'. and this creates some additional complication at a replication fork, which consequently on one strand the template with polarity of 3 dash to 1 dash, and this replication is continuous with one on the other hand the template with polarity, that is 5 dash to 3 dash, and it is discontinuous. So the discontinuous synthesized fragment are later joined by an enzyme called DNA ligase. The DNA polymerase on their own cannot initiate the process of replication, so also the replication does not initiate randomly at any place in the DNA. So there is a different region in E. coli where the replication originates. So such regions are termed as the origin of replication, and this is because of the requirement of the origin of replication. There is a piece of DNA if needed to be propagated during recombinant DNA procedures, which are requiring a vector. A vector provides the origin of replication. So replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of a cell cycle, and the replication of DNA in cell division cycle should be highly coordinated. A failure in cell division after the DNA replication result in the polyploidy, a chromosomal anomaly. So you will learn in detail the nature of origin and the process occurring through it. So stay tuned for the next video in which we will discuss about the transcription, RNA and all the remaining chapter. So stay tuned for the next video. Till then, thanks for watching. Don't forget to like, share and subscribe BioMindscape.